in ice cube isolated from Bengkulu, Indonesia. Ibu Marthadillah.
Yes.
Okay, the time for you.
Okay, thank you for the time. I will present our paper entitled Total Coliform Detection and Pathogenic Characterization of Escherichia coli in Ice Cubes Isolated from Bengkulu, Indonesia. We come from Faculty of Medicine and Health Science, University of Bengkulu. Okay, next slide. Okay, based on the report of World Health Organization in 2020, almost 50% of the world's population lack of the safe sanitation management, which cause various diseases due to microorganism contamination transmitted by through the drinking water. So the contaminated water source, poor of personal hygiene, and also environmental sanitation can contribute to acute diarrhea in toddlers. Acute diarrhea is in the developing countries common disease, specifically in Indonesia. The second leading cause, the death of in toddler, is responsible for the death of 370,000 children in 2019. And the number of diarrhea cases in Bengkulu province, also known as the 10 most infectious disease. Next slide. Okay, so the common bacteria found as the main cause of diarrhea among toddlers are Escherichia coli, Vibrio cholerae, and etc. And the most dominant is, we know that Escherichia coli. So the contaminated ice cubes with pathogenic microorganism emerge due to the use of the raw water and contaminated ice pack for the ice cube production during delivery or cold storage. So to validate the water quality result when monitoring the fecal population, the number of E. coli analysis has commonly been used. Characterization of E. coli is an important indicator of fecal contamination to evaluate public health risk, especially in water quality monitoring. Okay, next slide. For the material and method section, I think we collected 17 ice cube samples were taken from several home industry production in Bengkulu city coastal area. And the presence of coliform was identified by multiple two fermentation method containing a presumptive, confirmed, and completed tests following the FDA bacterial analytic manual. And then we continue to identify the E. coli. The positive result from the validation tests were cultured on a selective media using methylene blue agar, and the typical colony with a greenish metallic color were determined as the total viable bacteria before being cultured on nutrient agar for microscopic identification through gram staining. Then the presence of E. coli colonies was characterized by biochemical tests based on the American Society for Microbiology protocol, including indole, methyl red, Fogus proscura, and citrate, or abbreviation is IMVIC, before the carbohydrate fermentation analysis. And we also conducted the hemolysis test on a blood agar medium for the microbiological examination. And then we continue the microbiological examination to the molecular examination. So we extract the bacterial genomic DNA from all E. coli isolates from the previous detection following the procedure of Prestomini genome DNA bacteria kit, then we amplified the 16S rRNA following the Gottap-Green master mix procedures. And then the positive result of the amplified, we continue by Sanger sequencing method. Then the sequence were validated from the genomic data on the National Center for Biotechnology Information, or NCBI, using the BLAST. And then we conducted the neighbor joining to apply for the reconstructing the phylogenetic tree and evaluated by the involving a bootstrap of 1,000 replication. Next slide. Okay, for the result, the first, the coliform was identified using the multiple two fermentation method through presumptive and validation test, which show 10 samples. 10 samples from 17, from the total 17, were contaminated by coliform bacteria. The positive result of both tests were indicated by the presence of gas formation in the Durham tubes and green metallic and pink color colony and EMBA plates, as we see in the picture. And from the 10 samples, positive samples, after validation, we calculate, next slide. Okay, we calculated the most probable number value according to the FDA bacterial analytic manual to determine the coliform concentration. Based on this calculation, the MPN value of 10 samples had a coliform level of greater than 3 MPN per gram. So we can see that the highest total average coliform was found in sample in J sample with 43 MPM, followed by the K sample and L sample, while the lowest is in 3 MPN per gram. Next slide. Okay, to confirm the E. coli characteristic, biochemical tests were performed further. From this test, we can see that E. coli bacteria are characterized as indole positive, methyl red positive, Voges prosperous negative, and citrate negative. Furthermore, in mannitol and in carbohydrate fermentation test, E. coli positively fermentation for glucose, lactose, maltose, mannitol, and sucrose. These conditions were identified due to the changing color of the panel from red to yellow with the presence of gas in the Durham tubes. Next slide. We also identified the microscopic of the bacteria. So 6 isolates suspected as E. coli positive in EMB media with green metallic color colonies and were identified microscopic by the gram staining, and the results show the bacteria had bacilli formed with red color. Next slide. Okay, from this study, our study reveals that the six positive E. coli colonies were observed, with the total of bacteria are so based on the microscopic observation and also the gram positive protocol. It showed that the ice cube have been contaminated with the E. coli bacteria. Next slide. Okay, so the total bacteria or total coliform and E. coli are the main indicator of fecal contamination or presence of enteric pathogen. In several outbreaks of waterborne disease, coliform and E. coli have been detected in drinking water. So in addition, it is also associated with bacteria, virus, or unknown agent, but a study showed that 88% presence of coliform in water sample in the outbreak. So the E. coli strain also involved in diarrhea disease is one of the most causing agent referred to diarrhenic E. coli. So their E. coli strain found in water is crucial to characterize. Next slide. Okay, this is the visualization of DNA amplification, and the amplicon were sequenced to determine the sequence, and it showed the amplicon with the positive for the 16S rRNA. Next slide. Okay, this is the analysis based on the blast end result. The isolates have a strong similarity level with the avian pathogenic E. coli strain 018 and the E. coli of strain 0157H7, which are included as pathogenic E. coli strain. Okay. And we can see from the percent of identity and the query cover. Next slide. Okay, from the phylogenetic tree picture indicate that the closely related species between the E. coli isolates from the ice cube, the 6 positive ice cube with the E. coli strain of 0157H7 as one of the SIGA toxin producing E. coli or STAC. And also it is the main strain of enterohemorrhagic E. coli. Next slide. Okay, we also it also showed that 97 and to 98% similarity of the E. coli sequence from the gene bank. Okay, as I say before. Next slide. Okay, so based on our present study, we conclude that 6 isolates of ice cube samples consumed by Kampung Nelayan Sejahtera community in Bengkulu City are highly contaminated by E. coli with and according to 16S rRNA amplification result, all isolates are also positive as E. coli bacteria, and with the similarity, high similarity of the E. coli pathogenic strain. So after we compare with the sequence in the gene bank, one of isolate is closely related to the avian pathogenic E. coli 018 or EPIC 018. Okay, this is the end of my presentation. Next slide, and thank you.
Okay, thank you. Thank you very much, Ibu Marthadillah, for very good presentations about the total coliform detections and pathogenic characterization of E. coli in ice cubes isolated from Bengkulu, Indonesia. And we kindly ask Dr. Halimah to comment one comments because of the time constraints. Thank you.
Okay, thank you, Mr. Lalu. This study very nice. pemeriksaan E. coli-nya sampai kepada tahap apa tadi? Tahap lebih lanjut ya. Jadi
Molecular
molekuler.
examination.
Iya. What advice do you give for user of ice cube? Jadi bagaimana saran Anda pada pengguna-pengguna peminum-peminum es yang sudah terkontaminasi? Oke, thank you.
Thank you very much, Dr. Halima, for the question. I think from the result of our study, it proved that the highly contaminated of the ice cube in this coastal area. So we can suggest that the people in this area to be careful with the consume the ice cube. And I think it is interest to further observe the clinical data on the diarrhea case in this area, and then we can analyze the etiology related to the most dominant causative bacteria and its relation to the personal hygiene of the community in this area. I think
That's great explanations, Ibu Marthadillah. That's right. I think for the next step, I think it should to be confirmed in the clinical study, right? So thanks once again, Ibu Dr. Marthadillah, for very good presentations and also for very good response to Dr. Halima. Thanks once again. And we move to the last presenter will be Ibu Sunita. She will present her study about the leukocyte differential count in patients with type 2 DM treated with diabetic vascular port exercise. But at this time, Ibu Sunita will present by, she already sent the video to the committee members.